everyone, and welcome back to our biology catch-up sessions. My name's Ellesse and I'm part of the award-winning STEM response team at the University of Wolverhampton. And today, in this session, we'll be going over some essential maths skills that you'll need in your further education for biology, whether this be A-levels or BTECs. And we're going to start off by just recapping averages and ranges. So this should spark a few clogs in your brain going back to your GCSEs. So we have our averages here, we have mode, we have median, mean and range. Now, just for those of you that aren't sure or want to recap, the mode is our most common value. Median is always a middle value. The mean is our true average, and the range is the difference between the highest number and the lowest number. Now we're going to run through with an example here, just with some feet. So if you look at our feet here, you'll see we have 1, 2, 3, 4, 5, 6, 7, 8, 9 feet on our diagram, and they're all measured from 15 centimetres up to 20 centimetres. And we're going to use this example to work out our mode, our median, our range, and our mean. So already the mode is done. We can see from our example that the most common foot length here is 19 centimetres. So we can see that by arranging our data into manageable and measurable scales. Moving on, we can then see our median, so the middle foot length. And this is the one directly in the middle, which in this example is 18 centimetres. We can then work out our mean. And to do this, we have to add up all the different data sets we have and then divide it by the amount of data we have. So in this case, we'll add up all the foot lengths divided by 9, which is our data set. And that gives us a mean of 18 centimetres. And finally, to work out our range, we just take away the highest number from the lowest number. So in this case, we take away 20 centimetres from 15 centimetres, which gives us a range of 5 centimetres. Moving further on your A-levels and BTECs, you might also need to learn a thing called standard deviation. And this just means how much the answer you've got deviates from the mean, so how accurate your data is. Moving on now to fractions and percentages. Fractions and percentages show numbers as a proportion. So you can see here we've got some examples of some canary birds. If you imagine the question is, show as a percentage how many canary birds have yellow feathers, we can have a look and we can see that out of 12 canaries, 6 have yellow feathers. We can write it as a fraction, so 6 over 12, and then we can reduce that down to 3 over 6, and finally reduce it down even further to 1 over 2, or half. And we all know half as a percentage is 50%. Using our second example, we can see we have 9 canaries out of 12 with yellow feathers. So as a fraction, that is 9 over 12, and again cancelling it down is 3 out of 4, which is 75%. So now I'm going to show you how to actually work out harder percentages. So to work out a number as a percentage, for example, what is 6 out of 10 as a percentage, you always divide the first number by the second and then times the answer by 100. So for example, what is 6 out of 10 as a percentage? You would divide 6 by 10 to give you a decimal of 0.6. Then you would times that decimal by 100, which would give you the answer as 60%. So here's a question. A canary produces a clutch of 12 offspring. 5 of them have yellow feathers, 4 have green feathers, and 3 have red feathers. Using what we've just said in the previous slide, what percentage of canaries have red feathers? You can pause this video here to work it out. So to work out the percentage, we have to divide the smaller number by the total number. So we have to divide 3 from 12, which gives us a decimal of 0.25. We then times that decimal by 100 to give us 25%. Now, if you want to find a percentage of a number, it's very similar to do as well. all you do is take the percentage, divide it by 100 to get a decimal again, and then you times that decimal by the number you're trying to find out. So, for example, 60% of 10 is what? We take the 60 and we divide it by 100 to give us 0.6. We then times 0.6 by 10, as it's in our question, which gives us the answer of 6. To put that into practice, 85% of canaries surveyed exhibited green feathers. The total number of birds in the survey were 60. So how many birds had green feathers? Again, pause this video here and press play when you think you've got the answer. So to work it out, we want to find out 85% of 60. So we do 85 divided by 100, which gives us 0.85. We then times 0.85 by 60, which gives us our percentage of 51. Now we're going to have a quick run through of some graphs that you'll need to use in your biology further studies. Some of the most common graphs you'll see in your studies are bar charts, histograms and scattergrams, and we're going to show you the differences between those graphs now. So bar charts are used when you have categorical data, and they're really easy to compare that data. This can be pure data like frequencies, or it can also be percentages too. If you're comparing two different categories on your bar charts, you can do so in two ways. You can either do so like the top example, where you have one on top of the other bar, or you can do it where the bars are next to each other. The example of the graph on the bottom is the one that's most commonly used when you're comparing two categories with each other. When you come to make a graph or a bar chart in particular, you must always consider adding these things. First of all, always include your title, which explains what your graph will be showing. You then need your x-axis, which is the horizontal axis, and that is where your independent variable goes. In this case, it's our labelled categories. The y-axis, or the vertical axis, is where your dependent variable goes. And this should be in clear increments, always starting from 0. These can go up in 10s, in 5s, in 2s. It doesn't matter as long as it's clear to read and the increments are always the same. I would put temperature, for example, on the y-axis. Also, please remember when you do your graphs, always label your axes with what the information they are trying to show, so no one will get confused. Next up, we have our histograms, and these are used when the independent variable is continuous data, not categorical data. They look a bit like bar charts, but they are different. Bar charts have gaps in between each category, whereas histograms have no spaces between the bars, as it's continuous data. And also, these bars can be different widths as well, as you work out how high the bar is going to be based on frequency density. Now, we're not going to cover frequency density today, as you can find this information in our maths examples in about 2 weeks' time. The last graph we're going to give an example of is scatter graphs. Scatter graphs, or scattergrams as they're also called, show the correlation between two variables. So in the example I have here, we have the amount of bees present during different temperatures. You can see on the x-axis I have my temperature, and on the y-axis I have the quantity of bees that I have sampled. So you can see on our example that the data shows there are more bees present in our sample when the temperature is lower, and when the temperature rises, there are less bees present. And this shows us that there's a correlation between temperature and amount of bees. And in this particular example, you can see there's a negative trend. So when it's hotter, less bees are out. You might get a few data points that don't fit the trend, and these are called outliers. Now, to see the correlation, you can either have a negative correlation, a positive correlation, or no correlation at all. If your correlation is negative, you can see that your line, your trend line, will slope down across the graph. If your trend line rises up across the graph, that is a positive correlation. And if you can't fit a line in there, so there's not a line of best fit there, then there is no correlation. But to figure out if your correlation is significant, you have to do something called statistics. So we're going to go through a few basic statistics that you will need in your biology sessions. The first being chi-squared, then t-test, Spearman's rank correlation, and finally the index of diversity. Again, these will just be some quick examples, as you can find more information in our maths statistics sessions. So, chi-squared compares your observed data results with the results that you expected to get. So it's used for categorical data. If you surveyed an area of woodland and you found 4 species of birds, you would assume that your expected values would be the same amount of species between each bird. So if we found 20 birds of 4 species, you would assume you would find 5 of each of those species. Obviously that never really happens, and this is why we use chi-squared, to see if there is a significant difference between the amount of species and the variety in there. Similarly, you can also use a t-test, and this tests the differences between the means. So like your chi-squared, when you're looking at the amount of expected results with your observed results, t-test is used when you have more than one data set. So you can compare the means across different data sets. For example, you might do a bird survey on a piece of forest and see what animals live there, how many species of each bird live there, and then do another survey in a different aspect of forest. This can then compare the 2 forests and show you if there's a difference between the 2 data sets that you have. Moving on to Spearman's rank. This is the test that compares correlation. It measures the correlation of your data and it ranks the variables that you have. So if we go back to our B example with those scatter graphs, this is a test that you would use to determine whether that correlation is significant or not. With your Spearman's test, it should give you an answer between -1 to 1. A minus number means that the correlation is negative, whereas a positive number means the correlation is positive. The further away that number is to 0, the stronger the correlation. So if your statistical test says that the value is 0.37, that would be a moderate positive correlation. Whereas if it shows minus 0.79, that would be a strong negative correlation. And finally, moving on to the index of diversity, which is very important in biology to test the diversity of the area you're working in. This tests the richness and the evenness of the numbers of species in your survey. So richness is the number of species per sample, whereas evenness is the abundance of those species. And all this test does is just works out if the area is diverse based on the number of species with the amount of individuals that you found. There is one thing though, depending on your exam body, depends on how you do this test, because they both give slightly different answers. If you are with AQA or Edexcel, you'll see you'll need a different formula as to whether you're with OCR. So always check what exam body you are with and make sure you use the correct formula. So that was just a really quick example of some of the essential math skills you'll need for biology. Again, if you need to go into any more detail with these, please join us for our math sessions where you can find more deeper information on graphs, statistics, and trigonometry, for example. Thank you all for watching.